Penile squamous cell carcinoma is a rare malignancy with distinct biological behaviors based on human papillomavirus status. p53 loss of function mutations are common in HPV-negative PSCC and are associated with poor prognosis. This study uses single-cell RNA sequencing to explore the tumor microenvironment of PSCC, stratifying by HPV and p53 mutation status to identify transcriptomic differences that may guide personalized therapy. To do this, the authors used single-cell RNA sequencing and T-cell receptor sequencing to obtain a comprehensive atlas of the cellular architecture of PSCC, and tissues were obtained from 6 HPV-positive p53 wild-type, 6 HPV-negative p53 wild-type, and 4 p53 loss-of-function PSCC samples, and 6 controls were included. Immunohistochemistry and HE stains confirmed the morphological context of the observed signatures, and prognostic differences between patient groups were validated in 541 PSCC patients using Kaplan-Meier survival estimates. Stratification by HPV and p53 mutation status revealed 4 groups. The first was HPV-positive p53 wild-type tumors, which showed upregulation of E2F pathways, which is indicative of increased proliferation. On the other hand, looking at p53 loss-of-function tumors, these displayed pathway changes linked to epithelial to mesenchymal transition, hypoxia, and angiogenesis, all which are associated with aggressive tumor behavior. Indeed, when looking at the prognostic differences in a cohort of 541 patients presenting with p53 loss-of-function tumors had significantly worse outcomes, with a 3-year cancer-specific mortality of 25% compared to comparable 13 and 10% in the other 2 groups. Next, using pseudotime analysis, 2 distinct trajectories for tumor cells were identified. Indeed, HPV-positive p53 wild-type tumors followed a normal epithelial differentiation pathway, whereas p53 loss-of-function tumors progressed towards a partial EMT state characterized by aggressive behavior and active proliferation. To this end, histological analysis confirmed p53 loss-of-function tumors had higher levels of tumor budding, a feature associated with poor prognosis and linked to partial EMT phenotype. Next, by focusing on the tumor microenvironment, it was noted that the immune landscape varied significantly between the groups. Indeed, HPV-positive p53 wild-type tumors had an abundance of natural killer cells, indicating an active innate immune response. On the other hand, p53 loss-of-function tumors showed an increased regulatory T-cell expression, contributing to an immune-evasive environment. By looking at the checkpoint molecules, HPV-negative p53 wild-type tumors expressed a high level of immune checkpoint molecules, suggesting that they may respond to immune checkpoint blockade therapies. In contrast, p53 loss-of-function tumors had a low T-cell clonality, indicating poor responsiveness to ICBs. To conclude, p53 loss-of-function tumors are associated with a poor prognosis and aggressive phenotype, irrespective of HPV status. However, these findings suggest that p53 loss-of-function tumors may benefit from targeted therapy in pathways associated with EMT and angiogenesis, while HPV-positive p53 wild-type tumors could respond to antibody-drug conjugates and immunotherapy. HPV-negative p53 wild-type tumors, characterized by immune checkpoint expression, may be amenable to immune checkpoint blockade.